Okay, for this pre-lab, I'm just going to run through the lab itself and explain maybe there are some changes that we'll need to make. If you'll notice, this is lab exercise 3, chemical composition of cells. We have discussed proteins, carbohydrates, lipids. Those are the 3 things we'll be working with. And if you will turn the page, you'll see they have here a glucose plus a glucose, and this is a dehydration reaction where you pull out the water, and of course we form maltose, and that's a dehydration. And if we wanted to break that maltose down, you just add the water back into that, which would be a hydrolysis, to release both of the glucose molecules. So that maltose disaccharide would be broken, and you would end up with 2 glucose molecules. So that would break that down. Okay, and then on the next page, what you're seeing here is a starch molecule. So we're going to test for starch, and if you can read this print, it says you need to get 5 clean test tubes and mark them at the 1 centimeter level and fill the first mark with water, and then add, it does say 5 drops of iodine solution. You only need 2 drops, and in fact on all 5 of these tubes you're only going to use the 2 drops of iodine. Then on tube 2, then it says you're going to add a starch suspension to that tube to the 1 centimeter mark, and then you're going to add the 2 drops of the iodine. Okay, and starch does test positive using iodine, so that should give you a blue-black color, which is a positive test. Now on tube 3, it says use onion juice, add a few drops of onion juice to the test tube, and then again add 2 drops of your iodine solution, and see if there's a color change. And if you get a blue-black color, then that would mean that starch is present, and that's onion juice, and onion juice really doesn't have any starch in that. And then on the 4th tube, you're adding, this is potato juice, which should, of course, potato is a starch, that should give us a blue-black color using the 2 drops of the iodine. And the last tube, you're adding glucose. Now glucose is not a starch, that is a monosaccharide. Starch is a polysaccharide, many sugars. So if you add the 2 drops of the iodine solution, you should not get a color change, at least not a blue-black, which is the positive test color for starch using iodine. So you do need to know what you're testing for. In this case would be starch. You need to know what the reagent is that you use to test for starch, which is iodine, and you need to know the positive test color, which is a blue-black color. Now if you'll notice in test tube number 1, we used water. Now in that case, this is what you call a control tube, meaning that we know water has no starch in it, and so therefore it gives us something to compare to. So we know that in tube 1 we're not going to get the blue-black color. That will not happen because obviously there is no starch in just pure water. So anytime you're using the tube that has the water in it, it is called a control tube, and it is for something that you can use to compare with that you know is not going to give you a positive test color. So that one again is the control tube. Alright, now if we move on over to the next page, on the conclusion page, you can read that and answer that for yourself. And then on the potato, what you'll need to do is if you'll notice there on the potato number 1, second line, it talks about a drop of water. Just put a drop of iodine there instead. It's called I2Ki. It's just the iodine solution. and then omit the number 2 altogether, and that will work out very well. Okay, and then with the onion, you will need to do that, and it does not say add a cover slip, but you will need to put a cover slip on that. Okay, now on the next page you'll see we're testing for sugar. So we're not testing for starch, which is a long chain of sugars. We're testing for sugar. And again, if you'll notice, tube 1, that's going to be filled with water. So that, of course, is going to be our control tube. And then you're going to add 5 drops of Benedict's reagent. Okay, now we know we're not going to get a positive test in that one. Now tube 2, you're using a glucose solution. Now glucose is a sugar, and of course you are using the Benedict's reagent, and that one will give you a positive test color because glucose is a sugar. Now these tubes we will need to put in a boiling water bath in order to get a positive test color. That test color will be an orangish color. Will tell us that we do have a positive test for that. And there are no changes that we need to make that. Just read that and follow that. So again, we need to know what we're testing for. In this case is sugar. What the reagent is that is used to test for sugar, that would be the Benedict's reagent, and what a positive test color, and that of course is going to be our orange color. Once again, that does require a boiling water bath in order for that to work. Okay, then we get over to the next page. They give you a place to record information and inform conclusions. They do address proteins. So proteins are made of individual monomers, which are called amino acids, and it's just long chains of amino acids that will be used to build a protein. So if we go over to the next page, there at the top you can see we have 2 amino acids, and in order of course to bond those together, we're going to pull the water out, which is a dehydration reaction, and we formed a bond between the 2 amino acids, and that would be our peptide bond. So right here you're seeing the peptide bond, and that will tell us we bonded those 2 amino acids together. Now if we wanted to break those 2 amino acids apart, all we have to do is add water back into that. Instead of remove the water, we're adding the water to break the 2 molecules apart. That of course is a hydrolysis reaction. So we're actually going the opposite direction, and we are breaking those 2 molecules back into the 2 amino acids. So we're going to test for the protein. Well, the first tube we are again using the distilled water. We're not using tap water for any of this since we are using it is a scientific test. We're not going to use our tap water. There are too many other particles in that, so we just use pure water. So that will be our control tube, and if you'll notice it says it says 5 drops of Biuret reagent. Well, Biuret reagent is the testing reagent, but in reality it consists of 2 things. So those 2 things will be 10 drops of potassium hydroxide, so that's 10 drops of KOH, and 3 drops of copper sulfate, and that's CuSO4. So the 10 drops of KOH and the 3 drops of CuSO4, that does make up the Biuret reagent. We keep those separate because if they're added together they break down very quickly, so it's good to keep them separate. Okay, so you can just forget about the 5 drops. What we're using is 10 drops of KOH and 3 drops of copper sulfate. And again, that is tube 1. We're not going to get any positive test for protein, which is what we're testing for, because we know that is the control tube. Now, second tube, again, we'll use instead of the 5 drops of Biuret, that again will be the 10 drops of KOH and the 3 drops of CuSO4. Albumin, which is egg white, that is a protein, that should give us a positive test. and a positive test will be a violet color. Some people call that purple, which is fine. The third tube, we're using pepsin. Pepsin is an enzyme, and enzymes are made of protein, so that should also give us a positive test. And then the last tube, we're using starch. Well, starch is not composed of amino acids, so it is not a protein, it is composed of sugars. And of course, we test for starch with iodine. We do not test for starch with the Biuret, which is the 10 drops of KOH and the 3 drops of CuSO4. So the tube 2 will give us a positive test, tube 3 will give us a positive, tube 4 will not give us a positive test. So once again, we're testing for, we need to know what we're testing for. In this case would be protein. We need to know what reagent is used. We do call it a Biuret reagent, and we need to know a positive test color would be either purple or would be the violet, and then you can fill in your table and answer your questions about that. The last test is a lipid test, and I will perform that for you. That's a very simple test, and don't worry about that. We'll discuss that in lab. Okay, and then you see there that we have emulsification of an oil. Now, if you'll notice, there's something called an emulsifier. Now, that emulsifier, if we can get a little closer to that, you can see that we have a polar end on that, meaning that it has positive, negative poles, just like water does. And then you see that we have a non-polar end on that. And so the non-polar end, of course, does not have positive or negative poles. Well, fat is totally non-polar. If you've ever put oil or fat or waxes in water, you notice that those just float on top of the water. They're not going to dissolve with the water. The water's polar. The fats are not going to be polar, so they will not dissolve. So what happens with emulsifiers? Emulsifiers include things like soaps, detergents, shampoos, dishwashing liquids. Those are all emulsifiers, but all emulsifiers have the polar end that will dissolve with water and the non-polar end which will dissolve into the fat, since the fat is non-polar. So what happens with all of the emulsifiers, again the detergents and soaps and shampoos, dishwashing liquids and so forth, those things really have, again, the polar end and the non-polar end. So the non-polar end, you can see, sticks into the fat, and the polar end will be away from the fat since it is polar, just like water is polar. Well, then, since we typically wash clothing in detergents that are emulsifiers, there's also water there. We wash dishes with water, we shampoo hair with water. What happens, the polar end, which is the kind of the circular structure, that's going to dissolve with the water. The non-polar end, since it's sticking in with the fat, will be pulled when the water is rinsed through the hair or off the body or through the clothing and so forth. And then what that does is break that fat into smaller droplets. So that's what emulsification actually does. So we do need to know what an emulsifier is, and you will perform a little emulsification, and that's there on the next page. And just there are no changes that need to be made with that, so you do just follow those directions as written. Okay, the only thing we do change is we are not going to do tube 3. So tube 3 you can omit, and then of course write your conclusions. Okay, the last page of this, we're not doing this at all, where it talks about, look at the adipose slide. Testing for foods, we're not doing that, so you won't have to worry about that. And then on the lab review, if you could just fill that out, and I will review it before you leave the lab. And again, this should take, it doesn't take long if you run these tests, particularly if you get maybe one person at your table to test for the carbohydrates, another person to run the test on the proteins, another person to run a test on the lipid, and then another person possibly to do the emulsification test. And then there was one section in there where the test on the lipids, you really don't need to do that. We'll do that for you. We'll show that to you. And then the other thing that we do in this lab, of course, is do a positive test for starch using the potato, and of course you'll get a negative test using the onion. Onion has a tremendous amount of sugar in that, so that certainly is not going to give you a positive test for starch. Okay, so that will be the total extent of this lab. Thanks.